In this video, we'll take a close look at the stages of interphase, which prepares the cell for division, and then walk through the stages of mitosis, where the actual division occurs. Understanding these processes is key to grasping how cells grow, replicate, and ensure the continuity of life. The cell cycle is the series of events that cells go through as they grow and divide. It's divided into two main phases: interphase and mitotic phase. Interphase is where the cell spends most of its life, carrying out everyday functions and preparing for division. Mitosis is the process of dividing the duplicated DNA into two identical daughter cells. Let's start by breaking down interphase into its three stages: G1, S, and G2 phases. The first stage of interphase is the G1 phase, or first gap phase. During G1, the cell grows in size and synthesizes various proteins and organelles. This phase is crucial because the cell is performing its routine functions, such as producing energy, synthesizing enzymes, and responding to signals from its environment. G1 also includes a checkpoint, where the cell assesses whether it has the necessary resources and energy to proceed to the next phase. If everything checks out, the cell moves on to DNA replication. Next is the S phase, or synthesis phase. This is where the cell duplicates its DNA, ensuring that each daughter cell will receive a complete set of chromosomes. The DNA is replicated with high fidelity, meaning that the genetic information is accurately copied. This phase is critical because errors during DNA replication can lead to mutations, which may cause diseases such as cancer. After the DNA is successfully replicated, the cell contains two copies of each chromosome called sister chromatids, which are joined at the centromere. The final stage of interphase is the G2 phase, or second gap phase. During G2, the cell continues to grow and produce the proteins and other components necessary for mitosis. This phase is essentially a final preparation before the cell enters mitosis. Another checkpoint occurs here, where the cell ensures that all the DNA has been replicated correctly and that it's ready to proceed to cell division. If any issues are detected, the cell can pause this checkpoint to repair the DNA, or in some cases, trigger programmed cell death, also known as apoptosis, to prevent the propagation of damaged cells. With interphase complete, the cell is fully prepared to enter mitosis, the process of dividing the duplicated DNA into two identical daughter cells. Mitosis is divided into four main stages: prophase, metaphase, anaphase, and telophase. Let's explore each of these stages in detail. The first stage of mitosis is prophase. During prophase, the chromatin, the combination of DNA and protein in the nucleus, condenses into distinct chromosomes. Each chromosome has already been replicated during the S phase and consists of two sister chromatids joined by a centromere. The mitotic spindle, a structure made of microtubules, begins to form and will play a crucial role in separating the chromatids. Additionally, the nuclear envelope that surrounds the nucleus starts to break down allowing the spindle fibers to interact with the chromosomes. Following prophase is metaphase, where chromosomes align along the cell's equatorial plane known as the metaphase plate. This alignment is critical because it ensures that each daughter cell will receive an identical set of chromosomes. The spindle fibers attach to the centromeres of the chromosomes, holding them in place and preparing them for separation. Next, we enter anaphase. During this stage, the centromeres split and the spindle fibers shorten, pulling the sister chromatids apart toward opposite poles of the cell. This separation ensures that each new cell will have an identical set of chromosomes. Anaphase is a precise and tightly regulated process. Errors here can lead to cells with an abnormal number of chromosomes, which can cause genetic disorders. The final stage of mitosis is telophase. In this stage, the separated chromosomes reach the opposite poles of the cell. The nuclear envelope reforms around each set of chromosomes, creating two distinct nuclei within the cell. The chromosomes begin to decondense back into chromatin, signaling the end of mitosis. Although mitosis is complete, the cell cycle concludes with cytokinesis, the process of dividing the cytoplasm to form two distinct daughter cells. In animal cells, this occurs through the pinching in of the cell membrane, a process known as cleavage. In plant cells, a cell plate forms along the center line of the cell, eventually developing into a new cell wall that separates the two daughter cells. Each daughter cell is now a fully functional, genetically identical copy of the original cell, ready to enter its own cell cycle. The cell cycle, with its carefully coordinated phases of interphase and mitosis, is fundamental to life. It's how organisms grow, repair tissues, and reproduce at the cellular level. The precision of these processes ensures that genetic information is accurately passed from one generation of cells to the next, maintaining the integrity of life.